Okay, so all right, so we're done with talking about how you make proteins from DNA. Now, in the remaining time, let's talk about. So here we have fertilization. We have an egg right here, and we have all the sperm fertilizing it. So how we all started this way, but how do you go from that and end up as a fully grown human adult? So how do you go from one cell to the billions of cells in your body? And not only how do we go from one cell to a fully developed human, and remember, like, are the cells in your eyes the same as on your skin, as your muscles, as your neurons? They're different types of cells. But the thing is that do you just grow to a certain point and grow no more cells after that point? Thing is that what if you get injury? Well, we do have a ability to heal certain wounds, right? So what we have here, she lost a bunch of epidermal cells and some like subcutaneous cells right here, but eventually she regenerated cells to repair that wound. So how do we replace lost cells? We lose cells all the time. So not only like how do we go from one cell to full human, but in case we lose some cells that can be replaced, how do we replace the cells? So cell division. So this is what you need to do. You need to not only multiply and generate multiple cells, but you also need to like make up and replace lost cells as well. And this is actually, I don't think there's a real good equivalent of this picture in OpenStax. If not, I have to kind of look around more. But this is from Martini, and it's in your slides as well. So what we have here is, or actually, I think I didn't upload the slides yet, but don't worry, it'll be up soon. So in figure, you have to kind of go up to chapter 28, and I think it does a good summary of mitosis and meiosis. I think chapter 3 does mention both, but doesn't really have a figure of both. So do I want you to know the difference between these right now? Well, to a certain extent. So mitosis and meiosis, so what I want you to establish is some vocabulary. A parent cell, just like a parent your parents generate you. A parent cell is a cell that's about to divide into two new cells. So this is the parent. And offspring are called daughter cells. And daughter cells are cells that result from cell division. So you go from one parent and generate two daughter cells. And notice that those two daughter cells, they're in terms, or actually I'll get to that real soon. So this is mitosis. So this is the parent cell. Again, this is the one that's going to divide into two daughter cells. Now notice that undergoing mitosis and yes, someone said correctly, Julie is correct, that mitosis generates all those cells. But the thing is that this parent cell, and look at these daughter cells. Now compare the parent cell to the daughter cells. What do you notice? They're pretty much identical. So this is one key feature of mitosis. Not only do you get two cells from one parent cell, but these two cells are identical to each other, and they're identical to the original parent cell. So pretty much the somatic cells in your body, that's how you generate more somatic cells. And remember, somatic cells are everything but the sex cells. So sex cells are, again, the egg in females and sperm in males. Now this is the opposite, or not, I shouldn't say opposite, but this is the other type of cell division, meiosis. What do you notice here? Okay, don't worry too much about the difference between meiosis 1 and meiosis 2 at this point. In PHYL 142, we go more into detail about this, but at this point, what I want you to compare are the parent cells and the daughter cells. So the parent cells and the daughter cells, what do you notice here? Well, let's compare them. Notice that compared to mitosis, remember you only had two daughter cells. In meiosis, what we end up after meiosis 2, what you have are four daughter cells. And notice what's different. Well, one, the daughter cells are actually slightly different from each other, and they also have half the amount of chromosomes. But it kind of makes sense. If you're generating a sperm cell and an egg cell, well, when you have two halves, it makes another whole, right? So the thing is that if you didn't do this, have half the amount of chromosomes, you have doubling of chromosomes every time you create a new fertilized egg. So this is why it's important to have the amount of chromosomes in a sex cell. But again, very general at this point, but I want you to kind of help give you a better picture of what's the difference between mitosis and meiosis. You're generating more cells, but notice that mitosis keeps things identical between parent and daughter, but in the meiosis, you start to generate daughters that have half the chromosomes, but they're also way different from the parent cells. So yes, both eggs and sperm are generated via meiosis. All right, so then they are both types of cell division. The main difference, mitosis, two identical daughter cells. And meiosis, you have four sex cells, but they have half the chromosomes as the parent. And they also tend to be different from each other due to something called recombination, but that's, I think, a more advanced topic for a later class or actually maybe next semester, if not one of your more advanced classes. Okay, so now we have to talk about the cell cycle. So mitosis is only one part of the cell cycle. And we have four main phases. Here we have G1, gap, or sometimes called growth 1 or gap 1, the S phase or synthesis phase, then G2, sometimes called growth 2 or gap 2, and then you have that famous mitosis and cytokinesis near the end. So this is the way I remember it. Some people, I think your book is, I think OpenStax says it's the gap 1 phase, but this is the way I like to think about what happens during each phase of the cell cycle. So one cell cycle means one division via one round of cell division. So I like to think of it as growth 1, because the thing is that if you had just had your cell and it just split itself without growing whatsoever, it's going to have half the amount of organelles, half the volume, so it's going to get half the overall size. So in order to divide into roughly identical cells, it actually needs to start to synthesize more organelles, more proteins, more cytoplasm. So the cell undergoes some sort of growth. And then S phase is very special, it synthesizes something. So it's not it's synthesizing proteins during this whole cell cycle, but it synthesizes something very special and important. It synthesizes DNA. So this is what the very important feature of S phase in a cell cycle. So what happens with DNA synthesis? Well, things that, remember, you don't want if you want the same instructions in each of your somatic cells in your body, you don't want to cause any alterations, because that would mean, okay, this cell is different from this cell. So the different parts of your body might end up looking different from each other if it kept on changing every single time you went under mitosis. So you want an exact copy from the parent cells to daughter cells in mitosis. How do you get exact copies? You need to copy the information exactly. So when you have DNA synthesis, what happens is that you unwind that DNA molecule, and then DNA polymerase comes along and uses base pairing to fill in the gaps. What do you notice between, so I'm here showing the old DNA molecule in this black backbone, and I just filled in base pairing, and what do you notice with the new nucleic acid molecules that have the yellow backbone? Notice that not only they're base paired, but between these two new strands of DNA, they're exactly the same. So this is why base pairing is very important. This is how you copy information exactly between DNA, or when you're doing DNA synthesis, or transferring that information from DNA to RNA. So again, this is why I make a big deal about nucleic acid base pairing. This is how you make new DNA molecules that are exact. This is how you copy that information from DNA to RNA. So again, A binds to T, C binds to G. Now, why is S phase necessary? Well, the thing is that what happens at the very end? You divide your cell into two. So if you didn't synthesize or make double the copies of DNA in a cell, what happens if you just take your existing DNA without making a new copy and just divide it? You would half the amount of DNA every time your cell divides. So S phase is necessary because, again, you have all those chromosomes, all that DNA. But if you didn't make a duplicate and then you divide it by half, you would just eventually keep on separating your DNA by half, by half, by half, by half. And what happens if you multiply halves until infinite? What's 1/2 to an infinite power? That would be 0, right? So your DNA would be torn up into halves until pretty much equivalent almost nothing. So this is why you have to keep on doubling it every time, because you're dividing by 2 every time you divide the cell. You have to multiply your DNA by 2 to counteract the division by 2. So again, this is why it's very important to have S phase. Now, after S phase, you have the G2 phase. And what's the difference between G1 and G2? Always comes up. So this is why I like to call it just growth 2 phase. I mean, you still need that. The thing is that your cell likes to double check its work, because say you make a duplicate of your DNA and there's problems, like maybe the DNA is damaged or there's some sort of inconsistencies or errors. This buys a little time before actually committing to mitosis. So just think of it as another growth phase at this point. And then you have mitosis. So yeah, what happens if an error occurs during the process of the cycle? So there's something, now I won't go too much into detail, but there's something called cell cycle checkpoints. The thing is that your cells they don't just go like full bore through the cell cycle. They actually have parts in the cell cycle where they say, hey, is the DNA intact? Are there errors in the DNA? Is there something else not ready in the cell? Is there something weird in the cell? So the thing is that your cells can actually stop the cell cycle if it senses something is wrong with the cellular machinery that's responsible for dividing and creating a new cell. And in extreme case, so if it's a minor error, it tries to repair it. But the cool thing is that your cells, if they think it's like a lost cause and there's too many things messed up in the cell, it can actually cause the cell to self-destruct. And we're actually going to get to that term, apoptosis, pretty quickly. Yep, so there's also proofreading. Yes, so the cool thing is that your cells have all these DNA repair enzymes that do proofreading to make sure that there's no errors in when you're making a copy of DNA. So mitosis is the part when you actually have cell division. So what you have here is the prophase. So difference, I think your OpenStack says pro-metaphase. Don't worry too much about the difference between prophase and pro-metaphase and metaphase. I concentrate more on the difference between prophase and metaphase. So prophase, what you notice is that that loose chromatin is now forming a more dense structure, those structures that look like these little fat X's right here. So these are chromosomes. So prophase, you have this what you call chromosome condensation. It's forming these more compact structures. Now metaphase, the main feature, easy way to tell a cell's in metaphase is meets in the middle. So I like to think of it, that's one thing I learned actually from my AP bio teacher all the way back in high school, many ages ago. But yeah, so metaphase, she always said like they meet in the middle. So metaphase meet in the middle. Then anaphase, they break apart. So that's what happens here. And I think her remark was like, they were meeting in the middle, but someone named Anna came along and broke them up. So that's the anaphase. They break up because of Anna. And then telophase, now they're broken up. Now they're on opposite sides of a cell. So now they're telephoning each other. So telophase is when they're already broken up and they're opposite sides, and you have these two new nuclei forming. So they're already telephoning each other now that they're long distance relationship. And cytokinesis, this is when it finally breaks apart and divides into, or pinches off. It's not like breaking apart an egg. What you actually have is like a protein ring kind of forming a loop and kind of pinches off until the two plasma membranes separate to get from each other. And centrioles, actually, what are these? So this is what you see here. They look like little mini churros, right? So these are actually send out these microtubules. And what I like to draw an analogy between these, like during anaphase, during prophase, they kind of have these attachments via microtubules to all of these chromosomes and chromatids. So what they do is like you kind of have like two arrays of like they're throwing out fishing lines, and then on opposite sides they're starting to reel each other in. So during anaphase, they're adding more tension until they separate, and they're drawing it to opposite sides of the cell. Okay, so again, don't worry too much about pro-metaphase, but I want you to know the prophase, metaphase, anaphase, telophase, and cytokinesis. All right, so a nice mnemonic. So interphase actually refers to everything before you enter cell division. So interphase is like everything before mitosis. So another mnemonic, I'll pass my anatomy test. Tells you all the phases in order. Again, for this class, don't worry too much about pro-metaphase. So another thing about the cell cycle, and when you see these images like this in the Martini version, what you often see is that a common misconception is that mitosis is one of the longest phases. It's very complex. It has all these subphases, but look at the hours. Notice that G1 is typically the longest. S takes a while. G2 is relatively short, but mitosis right here, 1 to 3 hours. So mitosis is actually, though it's not 25% of the whole cell cycle, it's actually the shortest phase of the cell cycle. It's very complex, but all that complex phases happen in a very small amount of time. They're just showing it in a more expanded version so that it's more clear. Otherwise, it would be like all bunched together in a very thin slice of the pie. So again, when you look at these images, don't think mitosis is very long. It's very complex, but the actual time in terms of a cell cycle is relatively short. All right, so then, so we have G1, S phase, G2, mitosis. So then you have something called G0 phase. So sometimes it's called quiescence, or actually, so there's actually two terms for G0 phase. So things that when a cell isn't actively dividing, this is what we call G0. So if it's a cell that can divide, but isn't actively dividing, so it's like, I could divide, but I'm not going to. That's what you call quiescence. Whereas senescence is actually like if a cell divide, well, it's actually very important in terms of cancer biology. And don't worry, I italicized this one because it's a little more nuanced than just simple quiescence. But senescence is when a cell is pretty much done dividing and it can't really divide anymore. So it's still alive, but can't undergo another round of the cell cycle. So it's like a completely exited the cell cycle altogether, still alive, but just can't divide again anymore. So it's kind of like quiescence is like idling your car, if like the cell cycle is like your motor. Quiescence is kind of like idling and you're choosing not to move on and go forward, but you're just kind of like keeping things running. Whereas senescence is more like shutting down your car altogether, like you're just off the road. Yeah, so quiescence, like nerve cells, yeah. So cells that are, I don't think we have time for differentiation, but cells that are fully differentiated and cannot divide, they're still alive, but again, they can't divide anymore. So again, G0 is kind of exiting that cell cycle phase, and still, the cell's still alive, it's just not dividing. But the thing is that some cells can exit the quiescent stage, and say like if you lost, like the cells in your skin, if you're, I mean, they're always replacing each other. What's a better example? I'll think of a better example sometime. But things that sometimes when your cells need to replace each other, they can exit quiescence and enter the cell cycle again to replace lost cells. Okay, so then let's go to that word I alluded to earlier. So apoptosis, right? Or apoptosis. Sometimes there's an argument. I mean, my colleagues even argue, like, do you pronounce the second P? So just know how it's spelled. You're not being tested on pronunciation here. So what we have here are cells, but what's happening to them? So I'm going to show the process of apoptosis, right? So as they're glowing, what do you notice? That they're changing shape and they're also shrinking. So they're losing their shape. They went from these healthy cells that were very big and broad, and now they're shrinking into these tiny dead cells. And those that are very granular, very small. So what they're undergoing is cell death. But it's not just cells dying randomly, like say you have an injury or you burn cells. This is some cell, there's certain mechanisms or certain ways you can trigger cells to actually commit, or sometimes people like to use a more darker analogy. It's like cells committing suicide, but it's not due to like the cells not being able to handle what they're doing, or actually it is kind of like that. So the thing is that cells can trigger cell death due to certain mechanisms that are built in. It's kind of more like a self-destruct button. And things that, well, do you want that to happen at random? No, that means random things would die in your body. But the thing with apoptosis, sometimes you need cells to make room or get rid of cells that you don't need. So why do you need apoptosis? Well, it's the one thing is that when you're developing embryo, you have a tail. So I mean, or I don't think so unless you actually have a tail, I mean, the tail that you had as an embryo actually went under apoptosis while you were developing in the womb. Although remember earlier lecture where I showed that guy with the tail? So his tail actually didn't undergo apoptosis. At least that's why that's conjecture on my part. But another thing why you have apoptosis, well, notice what you have between the fingers and digits in this developing embryo. So this is why you typically do not have webbed fingers. I think, oh, I should take a picture, but actually there was a student that did have slightly webbed fingers. I mean, everyone has it to an extent. But the thing is that why don't you have extensive webbing like this person? Well, that's because their skin cells and between their two digits right here, they didn't undergo apoptosis, so it was kept from development. But another big reason why you need apoptosis, and I did refer to this a little earlier in the lecture, is that during cell division and the cell cycle, if too many mistakes occur in your during cell division and making a new DNA molecule, then it might cause so much alterations to DNA that your cells decide, hey, if I proceed with cell division and keep making copies of this error, it's going to perpetuate these errors and cause these errors to accumulate. So this is one of the underlying concepts behind cancer. If you cause alterations to your cell's DNA that prevent it from obeying the boundaries of where these cells are supposed to be, or stop dividing when there's enough cells, what we have here are cells dividing uncontrolled, and in fact, they're starting to grow so uncontrolled, they're starting to invade surrounding tissues and push out other cells and spread beyond where they're supposed to be. So if there's a lack of apoptosis or something goes wrong with apoptosis, that can lead to cancer. So what causes cancer? So the thing is that cancer, so some people do have genetic predisposition to cancer. Like I think, who's an example? Oh yeah, so like Angelina Jolie, she got a double mastectomy because she has a genetic predisposition to breast cancer. So that's about 5 to 10% of cases. Some people have a genetic predisposition to develop certain types of cancers, but the majority of cancers are actually due to DNA mutations. So it's due to some error in making a copy of DNA or some error in how you go from DNA to protein. That can cause cancer as well. So the thing is that, remember, I keep on bringing this up, nucleic acids contain information, right? So what do mutations do to the information in DNA? So here we have our mRNA molecule, and here we have our proteins. But what if you just change one letter? So in this example, we changed one letter. We just went from a cytosine to uracil. But remember that with those codons, oh, by the way, it always comes up, do you have to memorize which amino acid corresponds to which codon? No, not for this class. And even when I took Bio 265, I don't think we had to memorize because there's 64 different possible types of codons. And like, so that's a lot to memorize. I don't think that's something fair for our class. But what I'm getting at is that by changing that word, and changing just one letter in that word, that changes the amino acid. So sometimes you get lucky. Sometimes it actually doesn't change the amino acid, but this is something that can happen if you have a mutation in DNA. Mutation in DNA will be carried on to the mRNA and can potentially translate and carry on to adding the wrong amino acid to a protein. Okay, so let's talk about cancer again. So how does this relate to cancer? So actually this relates to radiation DNA. And let's see, I hope I'm saying her name right. But yeah, Charisse, oh, she has my favorite pack question to date. When I saw the initial question, I'm like, oh, are you serious? But she actually had a really thoughtful post, and she talked about cobalt-60 and gamma radiation. This is I love this question. And what does radiation do to DNA? That's why I kind of like, wow, this is a great question. This is a good time to teach everyone, or for everyone to learn about what radiation does to DNA. So what is radiation itself? So radiation is this is electromagnetic radiation. So even the stuff you're seeing, right, all the colors you're seeing right now, it is a form of electromagnetic radiation. This is what we call visible light. But as the wavelength gets shorter, you start to go from visible light to UV light to X-rays to gamma rays. So not only do these waves in this EM electromagnetic radiation get smaller between the intervals, they actually have more energy. So this is an interesting thing. So things like ultraviolet rays have more energy than the light we're seeing right now. X-rays have even more energy than that. And gamma rays have way more energy than X-rays in our visible light. So what happens with ultraviolet light? So we'll get to this in a bit when we cover chapter 5 and the integument. But ultraviolet light can actually cause those single letter changes in the nucleic acid. So here we have ultraviolet light, and it's changing a nucleic acid into a different form. Thing about this is that these have energy. Remember that I said chemical bonds have energy? So the thing about ultraviolet lights, or these higher energy forms of radiation, they can actually break bonds and rearrange things. Sometimes they can rearrange things that you could generate a completely different chemical altogether. All right, so then what we have here, so that was ultraviolet light. It can cause one thing to mutate. But if you have gamma rays, notice that compared to ultraviolet light, again, you're having increased energy as you go this way. So gamma rays compared to ultraviolet light, which is kind of like flicking things and kind of changing a few chemical bonds, gamma rays are super powerful. They're like taking a sledgehammer to your DNA or actually any molecule. So remember, DNA is made out of chemical bonds. But gamma rays, what they typically do is not just cause one mutation, they can actually cause overall breaks in DNA structure, and not just only your DNA, even your things like the collagen that makes up your skin or other proteins in your cells, they can also break those bonds as well. So gamma rays, super high energy that can break chemical bonds and cause things to break apart. So actually, this is very important for cancer therapy. Why? Because the thing is that and this kind of calls back to the cell cycle. If the cell makes too many errors in replicating its DNA, it can actually undergo apoptosis because it's like, okay, this DNA is too damaged to salvage. So it's kind of like if you took your textbook and threw it in a paper shredder and just broke it up into little pieces. Is it really worth it to paste all these pieces together and make a photocopy again? It's better just to throw the whole thing out, right? So that's what your cells can do. So if your cells experience too extensive damage to its DNA, it can actually undergo apoptosis and commit cell death. All right, so chemotherapy. This is another, now there's many types of general chemotherapy, but what do we have in this type of chemotherapy? Actually, what we have here is that this is a common chemotherapeutic drug called cisplatin. And what it does is kind of binds to the DNA structure. So it's kind of like so remember, you need to replicate your DNA in order to create a new copy. But this is kind of like throwing a big monkey wrench into the structure, into the whole DNA synthesis and replication machinery. So by introducing this molecule that binds to DNA, actually that causes errors in reproduction, and this causes the cell to undergo apoptosis. This is another chemotherapeutic agent, so it's called 5-fluorouracil. So it looks like uracil, but it's different. And the thing is that this kind of just like operates in a similar concept to cisplatin in that when you have replication and you have all these like, this is kind of like almost think of it like a poison nucleotide. So if you introduce this to a cell, it's going to cause that cell to die because you have this nucleotide that's not good for replication and can cause errors and can cause the cell to die. And then microtubules, remember, these are the type of filaments. Again, you have the microfilaments, you have the intermediate and the microtubules. So these are the ones that are actually sent out when you do mitosis. So you can actually mess up the process of mitosis by adding these sort of compounds that kind of prevent these microtubules from forming. So why am I showing all of this? This is actually pretty advanced, these images, but part of the underlying theory behind chemotherapy is that you can throw drugs that mess up the whole cell cycle and DNA replication and cell division and mitosis process to actually kind of prevent cancer cells from dividing over and over again. But this is also a big reason why cancer and chemotherapy has a lot of side effects. You're killing cancer cells, but you're also killing healthy cells that are normal healthy cells that need to divide. So this is why for people who have general chemotherapy, why does their hair fall out? Well, you have all these cells that are generating the hair follicles. So if you kill those cells that are dividing, your hair is going to fall out. Why do they have trouble swallowing things or trouble digesting things? Thing is that all the cells that line what you call your gastrointestinal tract or GI tract, they are always constantly repairing themselves and dividing. So if you add chemotherapy, which is killing the cancer, but also killing healthy normal cells, then that also causes a lot of the side effects. And this is and your immune cells, they're always dividing as well. So this is why people undergoing chemotherapy and cancer patients have to be really careful when they go out, because if they're having this chemotherapeutic agents killing cancer cells, but also their immune cells, then they have lower immunity. But why do we go with chemotherapy if it's so bad? In theory, and for people who are responsive to chemotherapy, chemotherapy kills, since cancer cells divide faster than normal cells, when you have general chemotherapy, like the drugs I'm showing here, they tend to kill the cells that are dividing the fastest. So this is why You kill most of the cancer cells, but you do have the casualties from all those normal cells as well. Okay, and I think, ooh, we're out of time. So cell differentiation, and it sounds like things worked out in terms of Twitch. So I probably will continue on Twitch on Monday. So full recovery after chemo. So that depend. Yeah, that's the other thing about chemo. It's kind of, it's a, I would say it's like, that's the real tough thing. And actually, it's always a hot topic in chemo. And is how do we target the cancer cells itself? But people like I have a friend. She had breast cancer and she recovered from chemo. But one interesting thing about her hair is that she recovered. She's in remission for like 5 years, but her hair actually became curly. Like she used to have this very like thick wavy hair, but now it's like she has this very very tight curls. And she says like, and anecdotally that actually changes. So when her hair follicles regrew, but they changed a bit, and she calls it the chemo curl. Apparently, some of her other girlfriends who and fellow survivors, they had changes in their hair as well. So chemo yeah, so that's why sometimes you do recover, but some things can change. And actually, as cancer and chemotherapy treatments improve, and we're seeing kind of like long-term survivors and seeing like, hey, does that kind of affect their long-term health, or what about the long-term effects of chemo, or like for testicular cancer patients. So what they often do is like they're worried about when they take chemotherapy, if that will affect their dividing the stem cells of sperm cells that are in their body. So what often happens is that they actually donate or actually cryopreserve. They have a sperm sample of their sperm before they undergo chemotherapy, put it on liquid nitrogen, and save it until they want to have, if they decide to have children later on, because they're worried about if the chemotherapy will actually affect the DNA when they of theirs that makes or actually affect the DNA of the spermatogonia, which are the stem cells of sperm, that will eventually make their sperm later on. So that's long-term effects of chemotherapy. It's it's an ongoing topic, and I think what was it? Yeah, that's one of the big questions and topics of research. Can we make chemotherapy more targeted to kill cancer cells? And how do we limit the damage of chemotherapy? And what's another example? Oh, thing is that your heart does have some, so the heart has a limited ability to regenerate and repair itself. So what we notice with one common form of chemotherapy called doxorubicin is that it actually causes, if these people, their cancer is remission, but thanks to this like very powerful chemotherapeutic agent, they actually have long-term heart problems. But it's like, okay, so are they going to die from, I mean, if you don't give them the chemotherapy, they're going to die from that cancer. But one of the side effects of doxorubicin is that it causes long-term damage to their heart. So they survive the cancer, but at the same time, now their lifespan is somewhat or shortened by a certain amount of years because they accumulate minor heart damage over the course of chemotherapy. But very good question. Okay, so we're all done, but looks like Twitch is working out. So I'll send out another announcement Monday morning, but I will continue on Twitch on Monday morning. The lectures will be posted on YouTube. Again, wait 24 hours because YouTube takes some time. And my Wi-Fi connection is pretty slow. So, all right. So see everyone on Monday. Enjoy your weekend. Stay safe.